Hello and welcome to our podcast Pager. I'm George Milner.
And I'm Stan Dale.
Tune in to hear us talk with some amazing people about the subjects changing medicine today.
From stem cells to mapping the brain and medical ethics, we hope to give you a glimpse of the bright future ahead through discussions with some of the people making it happen. Hello, and welcome to one of the first episodes of the Pager podcast. This episode features Doug Melton, a Harvard biologist and a leader in the field of stem cell research. Doug's lab is focused on finding a cure for diabetes. They plan to replace the destroyed pancreatic beta cells with pancreatic beta cells derived from stem cells. In this episode, he gives us his insight into how scientists can go about finding good questions to answer, and the different motivations which different scientists have. We also talk about what first interested him in a career in science, and what questions in biology still fascinate him at this point in his career. Finally, we also get into some of the nuts and bolts about the problems which still need to be solved in the quest to cure diabetes. Without further ado, I give you the Pager interview with Doug Melton. 3, 2, 1, and we're live. Today I'm joined by Dr. Melton. Thank you for joining us today.
Glad to be here.
How are you doing?
Very good.
So I was thinking, just to kick us off, could you give us a bit of a background in what your lab does here at the Stem Cell Institute, Harvard Stem Cell and Regenerative Biology Department?
Sure, glad to do that. We have a kind of straightforward, simple project, which is the idea that diabetics have lost the ability to make their own insulin, and rather than inject insulin with a syringe or a pump, we want to use nature's solution, which is to put the pancreatic beta cell, the cell that reads blood sugars and secretes insulin, back into the patients. So it's a really straightforward idea. Use a pancreatic beta cell to cure the disease. It sounds simple, but it takes longer than I thought, but we're pretty far along that road.
So it's a regenerative solution to diabetes that you're pursuing?
Yeah, the idea would be that we would put back pancreatic beta cells, and then the patient would no longer have to check their blood sugars nor inject insulin, because the beta cell does both of those things very efficiently.
These cells essentially will take over the role of regulating the blood glucose and producing insulin.
Yeah, and I think it's worth remembering that this would also change the life of the patient, because they wouldn't constantly be managing their blood glucose and insulin injections and worrying about what they eat and worrying about how much exercise they do or whether they'll have a so-called low blood sugar in the evening. It would really be a solution to the disease.
Yes, I suppose even now with insulin as a therapy, type 1 diabetes as a chronic disease can be very debilitating, and people still do die from it, don't they?
They do. They not only die from it, even if they aren't that unfortunate, if they just have to live with it, it can control their life. Because as I say, everything they do, what they eat for breakfast, what they have for lunch, whether they do exercise or not, all of that has to be managed and monitored quite carefully.
Now these beta cells, they're derived from stem cells, is that correct? Embryonic stem cells, induced pluripotent stem cells.
Yes, about, I don't know, must be about 10 to 15 years ago, we set out on the simple idea that an embryonic or an induced pluripotent stem cell can make any cell in the body. And so we said, could we turn that into a beta cell? I think at the time I thought that would take maybe 3 to 5 years. It took closer to 10 to 12 years. But I'm glad to say about 4 years ago we managed to do it. So we can now make fully functional human beta cells that are like newborn baby beta cells, really very healthy, by directing human stem cells to differentiate in culture.
Which is something which for a long time people would say was not something could be done in vitro.
Yeah, that's a good point. I mean, I think there were reasonable objections. If you think about how cells normally develop in the body, they have other cells sending them signals, they have a blood supply, and we were trying to do this all without those things and without a kind of three-dimensional architecture in a tissue culture setting. And so I think the people who thought it wouldn't work had reasonable objections. On the other hand, until you try, you don't know. And so I'm glad we gave it our all.
So currently, where the lab is at right now, your people are working on improving the protocol for differentiating these stem cells into beta cells. And also, how do we transplant essentially allogenic cells which are not from a patient, into somebody without those cells also being rejected by the immune system? Is that right?
Right. Yes, you've raised two good points. One is, could we make an improvement in the protocol that is, if you really wanted to have complete mastery over it, you might be able to make 100% beta cells. But in fact, what we'd like is a composition that includes beta cells that make insulin, and also another endocrine cell, the alpha cell, which makes glucagon, the counter-regulatory hormone. So that the nice idea here would be, can you make a kind of designer or bespoke islet? Can you decide what cells are in it? And then maybe even make those cells more robust, say resistant to hypoxia or longer lived. I don't know how long that will take, but that's now within our sights, is to genetically modify the cells and the protocol to make these really ideal islets. Then you said correctly that even if we made the ideal islet, we have the problem of if you transplant them into any patient, they would be rejected. And certainly they would be rejected by a type 1 or a juvenile diabetic because they have an autoimmune problem. In a sense, it doesn't matter if we talk about type 1 or type 2. Anyone who would receive the cells would reject them because they're not self. There are two ways we're thinking about and pursuing to protect them. One is not biological, it's just physical. Can you get an encapsulation device, a kind of, I usually describe it as a tea bag, into which you'd put the cells. The immune cells then can't go in and kill them, but insulin and glucose can get across the tea bag and treat the disease. I think a better long-term solution is to genetically modify the cells to make them opaque, or at least difficult for the immune system to recognize. In the absolute ideal case, you would say the cells couldn't be recognized by the immune system. I don't know if that's possible, but if we can just reduce the immune attack, maybe use some systemic immunosuppressants. There are many examples of cells which a host receives and eventually kind of gets used to, won't reject. So that is our goal now. To put it more succinctly, we're trying to genetically modify the cells so they can be put into any patient.
And these cells, which you might describe as universal cells because they can be put into anyone, I suppose those would also have applications beyond type 1 diabetes. If you worked out the sort of gene edits you wanted to make in a cells to make them opaque, that could be transferable to some extent at least, to sort of heart cells, cardiomyocytes, other different kinds of cells that might be transplanted.
No, that's a very good point. They could be used for other kinds of cells, including like dopaminergic neurons or cardiomyocytes. I like endocrine cells because they're autoregulatory. And what I mean by that is for other cell types, you not only have to put them probably back into the organ, that is the brain or the heart, but you have to put the right amount. The nice thing you could say about endocrine cells is since they autoregulate, it's almost impossible to overdose the patient. So you could put in an excess number. And then once, say, in the case of insulin or glucagon, the blood sugars are normalized, those extra cells are just there as a spare reservoir. They're not going to hurt anything.
Yeah, okay, that actually is a very good point for why beta cells are such a good cell to be thinking about in terms of applying this technology.
Yeah, you remind me there's another good thing, if you can think of good when you talk about treating a disease. But another good thing is it's known from many years of treating diabetes now that insulin can be injected anywhere in the body, pretty much. People inject it around their tummy and their buttocks and their arm. And obviously, if you're talking about cardiomyocytes or dopaminergic neurons for Parkinson's, you don't put those anywhere in the body. They have to go back in the relevant organ, the heart or the brain.
Yeah, absolutely. Switching gears slightly, one of the things I wanted to get your 2 cents on is how do scientists go about choosing interesting questions to answer? I suppose, would you be able to give us some background on how you became interested in science in the first place?
Well, certainly it's easy for me to begin with my own background, although I don't like talking about myself so much. But if I reflect on what got me interested in science, it wasn't medicine. It wasn't treating people. It was really kind of the wonders of nature. And I think of two things years ago. One, when I first heard about limb regeneration in salamanders, I just couldn't stop thinking about that. I thought that was so interesting. And as an undergraduate, I was lucky to work in a laboratory, a fellow named David Stokam, who worked on salamander limb regeneration. And I still find that problem interesting. Even now, 30 years later, we still have a small project here in the lab working on it. And I think that's the kind of thing that is fascinating. Why can a, quote, lowly salamander regenerate its lower jaw, part of its heart, all 4 of its limbs, its tail? And I don't need to remind us that we can't do any of those things.
No.
Of course, we don't have a tail, but we can't regenerate our limbs. And then while I was doing that as an undergraduate, I read a Scientific American article, which was about an experiment that to this day I find just wondrous, because it was simple in design and execution and answered an important question. And that was a Scientific American article by John Gurdon on cloning frogs. And the article nicely described the puzzle of how do cells become different, and then a test, an experimental test, which is one of the things I like about science, is you can do an experiment to answer a question, an experimental test of taking the nucleus and all the genes from a fully specialized cell, a skin cell, and taking that nucleus, which has all the genes, and transferring it by injection into an enucleated egg, and then making another frog. And even to this day, I teach that experiment to undergraduates. It's such a wonderfully nice experiment. And by enormously good fortune, I was able to then find a way to go to England for a degree and work with John Gurdon. So I don't know if that's the right answer to your question, but that's my own path of how projects were selected, how I got interested in really developmental biology.
I think it's very interesting that those same things which captured your attention when you were an undergrad, when you were younger, you still have side projects in the lab working on them. You still teach them to undergrads.
Yeah,
that's
it's fun.
Yeah,
it's one of the nice things about science. In fact, one of the things I like to do, I occasionally talk to even younger students here, is to show them an egg of a salamander and the egg of a frog, which are pretty much indistinguishable. They both have a jelly coater on them. They're about the same size. They're from the same pond. And just ask the youngsters, how does that egg know whether it should make an animal with a tail or without a tail? How is that decided? I think that, in essence, is the problem of developmental biology. How do genes control body form?
Well, I suppose whilst you were at Cambridge, you were exposed to people who ended up doing very interesting things in developmental biology. How much do you think, generally speaking, academic advisors shape the way that young scientists view the world and how they troubleshoot questions?
I think they have an enormous effect, both in terms of deciding what questions should be asked, what's worth doing, but also the style and the approach to scientific problems. I am forever grateful to John Gurdon, who seemed to me to have a special talent for asking simple questions. And I don't mean simple-minded, but being able to define a question in a way where you could get an answer on an important problem. And in my own case, that was a time in my life when I was actually toying with the idea of a different career path. I actually went to Cambridge to do a degree in history and philosophy of science. And there were wonderful teachers there. But one of the things I learned when I was there is that I was unlikely ever to do anything really original or important in philosophy. I would spend my life, how would I put it, kind of commenting on what great thinkers really thought about.
Sort of arguments which had already been hashed out.
I still find that interesting, and I teach a course that touches on those things and go to seminars in the philosophy department from time to time. But I think I made the right decision. I was never going to do anything really important or original. And at that time, when I met John Gurdon, I was just impressed by the possibility that one could do something really interesting and potentially important in experimental science. So that was a kind of turning point in my life. And to answer your question, John Gurdon had a really defining effect on the path of my life.
So what motivated you to get involved in, was there anything specifically about diabetes which made you interested in pursuing this as sort of a research interest?
Yeah, well, that's a big and important question, is how do people choose research projects? And I think there are all kinds of reasons for it. One can look at big problems in society, or just something that one finds fascinating and interesting. In my own case, which is not that uncommon, my young son, my 6-month-old son, was diagnosed with diabetes. And as you can appreciate, at that age, it's an extremely challenging disease to deal with, because you can't ask the patient, how do you feel, how are your blood sugars? So you can imagine with a little baby, my wife is having to prick not just his fingers, but his the heel of his foot to measure his blood sugar. And Gail was up many evenings for years checking his blood sugar in the middle of the night. And so what I did, I think, is what any parent would do. I mean, you say, what am I going to do about this? And I was in retrospect, in a good position to change my career path. Before then, I'd been working on very basic problems in how frogs develop, how their body axis is made, and embryonic induction. But I was extremely fortunate in two ways. One, I was at a very good university here at Harvard with wonderful students. And secondly, I had generous support from a philanthropy called the Howard Hughes Medical Institute. And so I didn't really have to ask permission to change my career path. I just started working on how beta cells develop. But frankly, what my wife Gail had to do was harder than what I had to do. I was already working in developmental biology. Gail had to change her entire life, and as I tease her sometimes, I say she had to become Sam's pancreas, which in effect she did. And then our family's commitment to the disease was, I don't know how you'd say it, redoubled about 10 years later when my daughter, who was 14 at that time, came down with type 1 diabetes. You can imagine that's doubly annoying. It didn't change what we were doing. It just made it even more important.
Double down, so to speak.
Double down. That's a good way to put it. That's right.
So it must be interesting for you as a PI. You have a lot of postdocs and PhD students coming through the lab. You meet a lot of people, seeing a diversity of reasons why people get involved in science. Do you feel like there are some motivations which are more compelling than others? Yours is obviously very personal and very powerful to hear.
It's a good question. I don't know what is the quote right motivation. I see all kinds of different motivations among students. If I were going to give any comment on it, I would say try to pick a problem that is both interesting and important, because it won't be easy to solve it if you pick a big problem, and it better be interesting to you and others, and why not make it important as well? But important can be related to asking and answering questions about nature, or things that are medically important. I don't draw a distinction or give one more value over the other. Fortunately for me, you could say making pancreatic beta cells and finding a way to protect them is both interesting and important. So it is a nice conjunction of the two. In other words, I wouldn't work on anything that was unimportant. I don't think anyone should do that.
So the interest gets you through the research, and the importance makes it important that that happen, I suppose. Yeah.
And we can't forget that no matter where you're doing science, most science is expensive and depends on the generosity either of philanthropists or the taxpayers, and so it should have some relevance.
To sort of broader society, I suppose.
Yeah. I think in this period now, we might be entering a period where people are forgetting the value of asking and answering questions about our place in the universe or how nature works. And I hope that's never lost from our universities. I don't think we should only do things that are medically important.
Well, I was curious to ask you, you said you wouldn't work on something which you didn't think was important. What do you think about the idea of pursuing knowledge for knowledge's sake, answering questions which might not directly impact us?
I think that's essential. If we couldn't do that anymore, it would be no fun to be human, I would say. And
Agreed.
In fact, you remind me that in recent years I've taken an interest in a little animal that many know about called a tardigrade, sometimes called a water bear. And as I understand it, this tiny little creature is quite amazing. It can be boiled, it can be frozen, it can survive 10,000 times the amount of irradiation than human cells. So that just makes one smile to think, how does that little super animal do that? How does it keep its double helix together after you boil it and things like that? So I'm delighted that there are people working on this and cheering them on from the sidelines. That's why I like science.
Do you think taking a broad interest in science, say like biology, you're talking about different kinds of animals, is that useful? Like, do you end up finding things or getting ideas from things which you otherwise might not see if you were just focused, say, on treating diseases in humans?
Yes, it's certainly useful in terms of the end goal, but we shouldn't forget that it's also more
just
fun.
Enjoyable. Yeah, that sort of was the original point. Yeah.
When you have a team of people, first of all, I don't know any important thing in science. Well, there are probably a few things, but there's hardly anything that's been done in science by one person. So teamwork is essential. And if it's not fun to go to work with people and talk about other ideas, you just won't want to go. You won't want to get out of bed. So I like the fact that in my lab and in other labs in the university, there's a kind of breadth of investigation. And there are people next door on another floor who find different things interesting. I think that's what makes university life so much fun.
What do you think the sort of the biggest issues are when it comes to generating universal cells? What are the things that are stopping us from just making gene edits, differentiating those cells, and chucking them in?
I'd say two things about that. It's first a fascinating subject to think about, could you make a cell that you could put into any other person? First thing to say about that is when you talk to people who are knowledgeable in the field, much more so than I, the general reaction is you won't be able to do it, that our immune system has evolved not with one or two but multiple layers of distinguishing self from non-self. And they could be right, but they could also be wrong. So let's take the positive view that we will figure out a way to do that. I see really only one problem with that, which is if you made a cell that truly could not be recognized by the patient's or the host's immune system, and then if rarely that cell mutated and became tumorigenic or was going to cause a cancer, you have to be able to get rid of it. And we've just said that the body can't use its normal immunological mechanisms to rid it. So my reflexive response to that is, no problem, we'll put a suicide gene or two inside. And so I think this is a really exciting area. I'm glad you and others are interested in it. I think it has enormous potential to teach us a lot about the immune system, which is an interesting puzzle, and also enormous medical implications.
What do you mean by suicide gene?
A suicide gene, maybe I shouldn't use that word. It's a gene which, when you give the patient a chemical, a pill, it acts only on those cells which have that gene and causes them to self-destruct, to do what biologists call apoptosis, to kill itself. And it shouldn't harm any other cells. These things have been demonstrated to work in mice, and I think they likely can work in humans.
So those chemicals would be chemotherapy agents, other agents?
They could be really something quite simple, yes. Like,
really?
Well, drugs that are now used that have that effect, called an inductive effect, would be a drug like tamoxifen or ganciclovir, that kind of thing. It's really fun to think about what if you could add cells to people and they would accept them as foreign without harmful. I was reading recently about these salamanders which take algae into their egg and then as adults have algae and chloroplasts in their skin. So that's a sort of fun thing to think about. It reminds me of a funny T-shirt a student wore in my lab many years ago. There's a young man who was prematurely bald, and he said, his T-shirt said, "It's not a bald spot, it's a solar panel for a sex machine." And it makes me think about, what if in your head you had chloroplasts and you had your own solar panel?
You had your own solar panel, yeah.
Now, of course, that's kind of a silly idea. But if you take it from that kind of cartoonish idea and ask, what are the limitations of the kinds of living things we could have in ourselves? Are there ways we could get energy? Are there ways we could protect ourselves from other invading viruses or other things? It's something we should think about.
The way we think about ourselves is so predicated on the way we just happen to have evolved, but that doesn't necessarily tell us everything we need to know about how a human could exist, if that makes sense.
It makes many people nervous to think about speeding evolution, but that's really what we're talking about. We're increasing variation to look for advantages. But if you think about it a different way, isn't that what modern medicine is? We don't say, well, bacterial infections are natural phenomena, let's just see who survives them. We give people antibiotics.
Exactly,
so,
yeah.
I think we're already accustomed to the idea that we're willing and able to manipulate the human form. And so let's think about how to do that. Of course, there are very important ethical, I would say religious and moral concerns, but those should just be discussed publicly rather than said, because these things are ethically challenging, we shouldn't do them.
Avoid them, yeah. Switching gears slightly, 10 years ago, a lot of people would have said, okay, developing functional beta cells in vitro from stem cells, like you said, it's not going to be possible. We can't recreate the extracellular environment in which these tissues normally develop in the human body. You persevered with this research even though there was a lot of doubt about that. What is it like to actually switch gears or turn around on an idea?
I think at one time in the recent past, with a student here, I thought we could find easily the genes that were activated in limb regeneration, which were missing in humans or mammals. That turns out to be a much more complicated problem than I appreciated at the time. Although I would defend the effort by saying sometimes it's good to be naive and, how would I say it, not know too much. You can often think of reasons not to do something, but I like the spirit of trying things. In fact, I'm remembering that about a year or two ago I showed at a group meeting this little video of something which just makes me smile to think of it. It was Elon Musk's effort to send a rocket ship into space and then have the bottom part of it return to Earth and land on a platform in the sea. And apparently they tried that for a long time and it didn't work. But seeing those the two bases of the rockets reverse their orientation and land on a football field-like platform in the sea just makes me smile. I think that was something where people said, you're never going to do that, and then they did it. So I like young people, I like anyone who says, I want to have a go at it. I'm not giving a full and serious answer to your question, because what you're really asking is, how do you decide when to kill a project? I don't know the answer to that. I don't know. We have killed projects, and I guess at some point you just get frustrated and you say, I don't have any good new ideas. I can't think of another way to approach this at the moment. But the thing that's so wonderful about science is you can ask a question and design an experiment to test it. And that's something that is not easily done in other spheres of human activity.
So do things often get sidelined with this idea they might be returned to and then just sort of left to die from there?
They do, and they also get sidelined maybe because a student moves on or loses interest. And so it's up to the individual to decide how far they want to push on this. I imagine you could ask the same question of successful investors in the stock market. How do they know when to bail?
Pull out, yeah.
And I don't know the answer to that. But I guess the ones that are good at it have some intuition about when is the right time.
Yeah. Can you see parallels between you and people in tech like Elon Musk, who are just interested in finding things out, curious about, sort of infinitely curious about questions which might not necessarily lead somewhere, but are worth asking just for the sake of it?
I like to think I see parallels with myself with most of the faculty here at Harvard who think about questions like what if or why not. I think that's what makes life fun. I think it's probably obvious by now that I'm a great believer in science as a way of knowing. I think if you ask what do we know about the world, almost all of the answers, in my view, come from scientific investigation. And the scientific revolution has really changed what it means to be human in that way. So I'm more puzzled by people who don't want to be scientists because it's so much fun.
Can you expand on what you mean by science has changed what it means to be human?
Yeah, I can say that in many years past, when people asked questions about why do we have infections, why is there starvation, why do we have seasons, the natural philosophies at the time were usually things you might call religious. That is, people thought there was something else controlling biological infections. Why do we make babies? Why does a boy or a girl form? Now we have scientific answers to that. I think you could say the modern version of this is people who think global climate change has nothing to do with human behavior. Science is the only possible solution to global climate change, in my opinion. And if one imagines there's going to be some intervention from a godlike creature, I'd say good luck with that idea.
If you were going to get involved in science now, looking at problems which present themselves outside of human biology, something like climate change is obviously a huge question, with a huge problem we're trying to tackle. We're going to have to tackle this century. Do you reckon you would get involved in climate science? Or?
That's a good question. I sometimes think if I were starting off as an undergraduate now, what would I do? I don't think I would do climate change. Partly for the reason you indicated is that
it's so cold in New England, or
no, it's that things don't change quickly enough for me. So if I were going to start off now, what would I do? I think I'm still in love with limb regeneration, but I'm going to give a different answer. Let's think about the main way we interact with the environment. It's by the air we breathe and the food we eat. And curiously, science has not been all that effective at understanding the relationship between what we eat and who we are. I mean, the evidence for this is if you look in any newspaper this month or next month, you'll see some new diet which is supposed to improve everything, make you healthier, make you fitter or whatever. So I would work on that problem, is the relationship between what we eat and our genes. And to make that more specific, I'm a big fan of genetically modified foods. I can imagine a time when you would eat foods that contained gene products or chemicals that stimulated your body's inherent capacity for regeneration. Dare I say, some different version of healthy aging. But there's no reason to not think about eating, say, sweet corn, which also stimulates your muscle satellite cells or makes your cognitive abilities improve. That might sound crazy, but I can't think of any reason that's not possible. So short answer to your question is, if I were starting off now, I think I would work on genetically modified food.
Are you interested in the relationship between diet and aging?
I'm interested in aging in the sense of healthy aging. If you ask most people, do you want to live for a very long time, they might say yes, but then you say, now imagine yourself at 85 and infirm and hard of hearing and losing your memory. Do you want to live another 50 years like that? Most people say no. So I really like the idea of healthy aging, that we would extend the healthy part of our life more so than extend the absolute
length,
number of years
How
that
long
you're
can we
alive.
stay healthy within a given lifetime?
And that's an interesting question. How long do you think a human could live? I think we don't know the answer to that. We do know in the last century, I think the number has nearly doubled. I don't actually know the numbers exactly, but I remember seeing once in Massachusetts, the life expectancy in 1920 was about 30 or 40 years old for people. I think I have that right. And so in a century, we pretty much doubled the lifespan of humans. Of course, we now know that many of the humans who are living a long time, unfortunately, are suffering from things that make life less enjoyable. If you can't hear, if you can't walk, if you can't remember, that's not the same thing. You're still human, of course, but it's not the same thing of being a 25-year-old youngster who's ready for anything.
I suppose you're like the fact that becomes quite philosophical again. Your
Yes.
biographical life might have ended, even if your biological one hasn't. And what is the meaning of life beyond being able to interact with people and make an effect on the world?
Yes, I'm not all that interested in this high-tech solution to that of using computers for your digital memory and being able to interact with someone who's physically passed away, but many of their mental characteristics are available on a computer. That doesn't interest me.
To me, that sounds very dystopian. I think we should stick with the cells.
That's
I'm
right.
with you on that one. Okay, switching gears back to sort of the regenerative stuff. We had a conversation about thymic cells and regenerating thymic cells. And it captured my interest, even though it didn't fully compute for me. So I was wondering, could you sort of elucidate what your interest is in regenerating thymus cells? What's the point of doing this?
Yeah, that's a good question. My idea, which I don't think is an original one, is we know that the thymus is the organ in humans which educates our immune cells for self versus non-self. And so if that's true, could one make thymic epithelia, as it's called, a thymic organ, in vitro, and it would have the two main cellular parts known of that, the medullary and the cortical epithelia, which instruct, as I say, white blood cells to recognize self from non-self. So if you could make that in vitro, you could now begin to get control over it and master it. And so you could now take a person's thymus and their as yet unformed immune cells and show it a target that you wanted it to get rid of. You can think of like a bloodhound where you show it the criminal's shirt and tell it to smell it. And so you would show it a virus or a bacterium and say, this is your enemy. Now go back into the patient, and when you see that, get rid of it.
So you would circulate somebody's peripheral blood through sort of an artificial organoid of these cells you'd constructed.
Yeah, I mean, I think even easier would be to take hematopoietic stem cells from the person, have them differentiate and educated about what's self and not self, or an enemy and not enemy, and then put them back. So it could give a permanent immunity to things.
Oh, okay. once you clarify, just to clarify, when you talk about educate, you're talking about molecules being presented to these cells to show them, okay, this cell is self versus this cell is non-self. Is that right?
It can be cellular, or it could even be harmful chemicals.
Your lab is focused so heavily on curing type 1 diabetes, and it's sort of, you've elucidated a bit how that's come to be your focus. Are you interested in preventing type 1 diabetes? Do you let postdocs explore projects which are related to that?
Well, maybe this is a defect in me. I don't think much about type 2 diabetes, nor do I think much about prevention of type 1. So I would say selfishly, I just want to find a cure for type 1. To defend myself, I would say that if you try to do everything, you probably can't do it. So I'm not trying to cure and prevent all types of diabetes. I'm glad others are thinking about that. For me, it's a big enough problem to just try to cure type 1, and that's what my focus is.
Focus, yeah. I suppose that must be important with a lab full of different people who have different ideas and interests. Let's keep everyone essentially working towards one goal.
Yeah, that's a good point, but I find I like to think that my lab is reasonably productive and successful, and I think one of the things I've always done is to allow people a significant amount of freedom to explore new ideas. In general, it's a good idea for a student or a postdoc to have at least 2 projects. One of them I know quite a bit about, and if the other one is exploratory on the causes of the disease, or say recently someone feels he's discovered an insulin-independent pathway to control blood sugar, and that really came from his own ideas thinking about a new method we developed. So I like having a lab which has some, let's say, novel and experimental things at the margins, but we have to keep our focus on the goal.
I'm wondering, how do you go about, I don't know if recruiting is the right word, how do you convince people that, how do you go about saying you should be here, you should be in the Melton lab?
Yeah, the fact is I don't do that. And that's because I think science is hard. People work many hours a week. And I don't want to convince anyone to do this, nor do I think I'm very motivational. They have to have within them a kind of desire, but you might say a fire in their belly to want to do this. And then my job is to provide them the resources and environment where they can do that. I think being at a top institution like Harvard also helps because we get applications from lots of students. I don't think I've ever really tried to convince a student to do this rather than something else. I tell them what interests me and what we're doing here. And if they find that interesting, I'm delighted to have them join our team.
Okay, yeah. So I suppose you're in a nice position where this is a highly desired lab for people to be working in, so that must help.
I think I don't know if it's a desirable lab, but certainly the problem, many people find the problem a nice conjunction of being both interesting and important, and it's also reasonable to think about how you might solve it.
Yeah.
Like sometimes I think about problems like autism. That's interesting and it's important, but I don't really have any ideas about if I said our goal is to solve autism. I'm not really sure what I would do about that. Whereas I think type 1 diabetes is somehow a problem which you can draw a circle around and say these are approaches where we can at least imagine a solution.
Yeah, that's a much better way of putting it, actually. So here in Cambridge, Massachusetts, this is really a hub for like biotech companies, people who are doing gene editing to solve all sorts of different problems and introduce new technologies. I'm interested in sort of what is the relationship between labs like this, which are pushing research and papers out, and people who want to commercialize that research, produce products. How does that work? Are there pitfalls and problems which are encountered? Conflicts of interest, maybe?
It's a very juicy subject, and it's an important one. If you think about what are the institutions that the public rightly thinks are involved in trying to find solutions to let's say their disease. We have, let's call it pharma and biotech on the commercial side. We have hospitals, which largely are supposed to treat patients, but also do research. And we have universities. Then we have funding agencies, philanthropic organizations, the National Institutes of Health, which would be like the MRC. I think society hasn't quite figured out how to make those things all work together easily. They work together, but not so easily. So there used to be, in my lifetime, a kind of strong view within the university that commercial things were somehow not to be done here, that commercial aspects were, how would I put it, let's say academics were like intellectual thoroughbreds, and the people who did commercial things were more like a plow animal, right? That you didn't think about products. I have a completely different view. First of all, the commercial thing is very difficult. Secondly, it's intellectually challenging, and it's as valuable and hard as anything academics do. I also would like to remind scientists that if you do want to help people, you have to get used to the word commercialization. Just having an idea and finding a solution doesn't do any good until you figure out how to produce it and market it, if you like, get it to people. I would say, what are the barriers to this? The one that almost always comes up quickly is conflicts of interest. And someone told me recently a nice expression I like, that if there's no conflict of interest, there's no interest. And I find that in many ways very true. And so the question is, how should society manage that? Certainly there's a conflict of interest if a physician is prescribing an experimental drug for a patient when that physician has a financial interest in demonstrating that that drug works. But because that extreme example rightly worries people, that doesn't mean that we should have academics without strong connections to biotech and commercial things. And in my own case, I've been involved in forming several companies, and I'm very actively involved now with one that's trying to cure type 1 diabetes. So I think that's a good thing. And the university has all kinds of rules and regulations for how that can be done.
Yes, so I imagine there are issues surrounding the intellectual property of different techniques and stuff like that. So how does that actually work? If a postdoc in your lab has discovered something useful for, say, a company you're working with, how do you go about bridging those two together?
Yes, well, that, as you may know, varies from country to country. So I'll give you two extreme examples that I know of. In this country, anything that's discovered in my lab is owned and managed by Harvard University and the Howard Hughes Medical Institute. We don't own any of that, so if we make an important discovery, the university files for a patent, a U.S. patent and a worldwide patent, and then license that technology to a company, and then the revenues for that come back to the university. That's then divided up to the university and the department and all of the, what you described as inventors, which would include the student. I was interested to learn recently that in Sweden, it's not like that at all. The inventor owns the patent. The institution doesn't have any ownership over it, and the inventor can do whatever they want with it. I frankly don't know what happens in England, in the UK. I don't know the situation there.
The Swedish example is strange to me, because one doesn't come up with things in a vacuum. You do it using the resources of a place, under the sort of guidance of people who are managing your work. So what do you think about
It's
that?
true.
To me, that sounds.
But let me
peculiar.
remind you of a strangeness that also fits into this, is that if you are a Harvard professor and you use the Harvard library and the Harvard computer to write a book, and then you publish the book, you get all of the royalties. Harvard gets nothing. So why is that different from scientific discovery?
Okay, well, on the surface, you're right.
That's right.
They're
So
similar.
I think that's a different path was called copyright, and historically copyright just went to the author.
Went to the author. But yeah, this is a good point. But if you're using a laptop or a library, that feels more fair game. A lab is, I feel like in the setting of a lab, it's even more
Really?
collaborative.
What's the difference between our chemical shelf and the library? The library is a resource which allows the author to draw a conclusion. Without the library, they couldn't produce the book.
Well, it's interesting you say that, considering so many resources now when it comes to
digital,
literature.
yes.
Yeah, are much more available sort of on the internet.
That's a good question.
You can't get, whatever chemicals or media you need from the internet.
Yeah, I don't have a solution or a proposal. I was just pointing out the sort of curiosity about how in the humanities your work is considered entirely your own, and in the sciences your work is considered to be entirely owned by the university.
Either way, it seems like there are inevitably going to be difficult things which need to be managed within that sort of process of bringing something.
And I think the two issues one should always keep in mind is financial interest can cloud behavior, and harming people, of course, is something that has to be avoided. And that's what makes medicine and biotechnology different from writing a book.
Dr. Melton, I think that's about all we have time for. Thank you very much for joining us on the episode.
Oh, thanks
Hope you
for
enjoyed
inviting me.
Yeah, thank you. Thank you for listening to the Pager Podcast. If you've enjoyed it, then head to Spotify or iTunes to hear more.
As ever, we'd love to hear what you think, and you can reach us via our Facebook page or by email. See you next time.